Hello guys, today we shall reveal the real secret behind using the Punnett squares in biology. Stay connected till end to get the full insight. We know that germ mother cells in males and females are diploid cells and they undergo duplication before meiosis. So in this case we have taken a single homologous pair from each male and female and we have labeled a single locus allele, that is for seed shape in plants. You can say pea plant, round and wrinkled. So we have not taken all the other chromosomes because of our convenience. When meiosis will complete, these 4 haploid pollens will be formed. Similarly, in case of female or gynoecium, there will be 4 ovules formed. As we can see that due to duplication, we have 2 types of pollens and 2 types of ovules, and capital R, smaller are repeating. So if we ignore these repeated ones, then we have only capital R and small r in pollens and capital R, small r in ovules. So now we have to check the possible combination of these pollens and ovules with each other. So what is the probability of our offsprings having the seed either round or wrinkled? So we simply take these chromosomes, alleles of single locus on either side of our Punnett square. So let's extract the alleles only so that we may feel convenience. Then we draw the combinations. Here is the capital R, small r, and capital R, small r, and similarly small r. So that is the possibility of next generation seeds, whether they will be round or wrinkled. So that is the basic use of a Punnett square for a single allele to predict its future in the offspring. So if we talk about the genotypic ratio here, we see that capital R are homozygous round, capital R, small r are heterozygous round. So with respect to genotype, we have 1:1 ratio. And with respect to phenotypic is the expression. So apparently we have 3 round and 1 wrinkled. So we can say that there is a 3:1 ratio. And if we talk in percentages, then we have 75% round possibility and 25% wrinkled possibility in the next generation. So that's the basic use of, and this kind of cross is termed as monohybrid cross because we have taken single allele present on a single locus in a chromosome. So if we talk about dihybrid cross, now we have taken 2 alleles present at 2 different loci on our chromosomes. So the possible combinations in the pollens or ovules are shown in the screen. You can see that there are 4 chromosomes with different combinations of these 2 alleles on the pollen side and 4 same type of combination because we have to take the maximum probability of both sides. So now we need a bigger table to draw or predict the possibility. Now we take just these chromosomes of eggs and pollens on either side of the Punnett square and let's extract the alleles for convenience so that we may make the combinations easily. Now we have to check the possibility in the offsprings, that how many offsprings will have now one possibility and how many will have the second possibility. Now if we combine these alleles, we can see that capital R and capital Y. Remember here that the similar alleles are paired together, not the different ones. So capital R will be paired with capital R and Y will be paired with the Y. In the next box, this combination will take place like this, and in the 4th one like this. So now you can easily draw these combinations. So let's move faster and draw all these possible combinations and fill the checkerboard completely. Now we have a complete checkerboard, and because we have taken 2 alleles, so our checkerboard is larger, and there are 16 possible combinations of the future generation. So let's jot out the round yellow ones in the table. So all these are round yellow because they have capital R, and capital R is dominant, so all the seeds having capital R will be round, and having capital Y will be yellow because capital Y is for yellow color and small y is for green color. So if we count them, we find round yellow 9 in this checkerboard out of 16. So similarly, round green, we find round green 3 in number, and wrinkled yellow are also 3 in number, and wrinkled green only 1. So we get the combination as ratio 9 3 3 1. So that is the phenotypic probability in the next generation, that 9 out of 16 will be possibly round yellow, and 3 out of 16 will be possibly round green, and 3 out of 16 will be wrinkled yellow, and there is a possibility of only 1 in 16 that will be wrinkled green. So that is the basic use of Punnett squares in biology to predict the genotypes and phenotypes of the next generation or offsprings. And that's why we say monohybrid cross, because we take a single allele, and dihybrid cross because we take 2 alleles at the same time and then check their possible combination in the future generation. Similarly, we can take 3 alleles at the same time, but the board will be much larger and much complicated, so we stick only to 2 alleles at a time. So Mendel named that monohybrid cross because in the process of gamete formation, alleles on 2 different chromosomes segregate from each other. So that's why he named that law of segregation. And similarly, alleles on same chromosome or different chromosomes independently assort from each other. On this observation, he stated the law of independent assortment. Hope you got the basic point. Thanks for watching.